Dean sir, shall we start? Dean sir,
Yes, ma'am.
shall we start?
Yes, ma'am. We can start. Huh?
Good afternoon, everyone. A very warm welcome to all the delegates, faculty members, participants, and esteemed guests joining us today. I am Suchita Mishra, Assistant Professor of School of Pharmacy, Galgotias University, and it is my privilege to host day 5 of the concluding day of the Faculty Development Program on International Drug Design, Delivery, and Development. Our commitment and active participation throughout these 5 days have been made this FDP truly meaningful, collaborative, and enriching. So, for today, our esteemed guest is Dr. Chu Yip Ling. It is an honor to introduce our distinguished keynote speaker for today, Associate Professor Dr. Chu Yip Ling. I hope I am pronouncing it right, ma'am. From the Faculty of Pharmaceutical Sciences, UCSI University, Malaysia. Dr. Chu holds a BSc in Biotechnology honors, PhD from Monash University, and a postgraduate diploma in tertiary teaching. He is also a chartered scientist and an active member of Royal Society of Biology, UK. Her research expertise is span across antioxidants, antimicrobial, and anti-inflammatory potential of tropical plants, herbal medicines for managing skin disorders, identification of plant-based bioactive compounds, advocacy of phytomedicines, and natural therapeutic alternatives. We are privileged to have her with us today to share our to uh, to share her knowledge on and global perspective for drug development and design and discovery. Now, I warmly invite Dr. Chu to begin her lecture, ma'am. The stage is all yours.
Okay, thank you. Good morning to everyone. Let me share my presentation slide for today. Chairperson, can you see my presentation slide?
Yes, ma'am. We can see your slide.
Okay, thank you so much. So good morning, everyone. I'm very happy to give my presentation today to everyone over here in this EFDP program. So today I would like to talk about the rationale of drug design targeting on MRSA membrane. So we will, in this talk, we will actually look at how the Cipirocalor has the antibacterial mechanisms on the MRSA. Okay, a little bit on the statistics of this drug-resistant infection. So what we can see that from this slide is that there is an increasing in the antibiotic-resistant cases since 2020, and it is expected to be increasing up to 2050. Compared to cancer, all this while, we know that this is the #1 killer that we have. But we are expecting that drug-resistant infection is going to be increasing, and it will be a common cause of death in the future. If you compare to with diabetes, diarrheal disease, road traffic accidents, measles, and tetanus, these are very low when you compare to the antibiotic resistant infection. So this also means that bacteria are developing their resistance towards antibiotic, and there is a reason why bacteria are developing the resistance is one of the reason is the overuse of the antibiotics, okay? And second one is misuse of antibiotics. So these are the two common reasons of why the bacteria are slowly developing the resistance towards the antibiotic nowadays. And from this statistics here, we found that the number of cases for antibiotic resistance is increasing year by year. Okay, so this is why that we are expecting to have up to 8.7 million of that is due to the antibiotic resistant strain. Okay, this is also one of the statistics that shows the prevalence of gentamicin resistance among Malaysian Staphylococcus aureus isolate back from 1990 to 2017. So we can see here in those different kind of centers, healthcare centers. We have a lot of the healthcare centers in Malaysia. Okay, we have government, we have those private institution, which is for healthcare center. There is a lot of cases of these antibiotic resistant cases. It happens every year. And from here, you can see that there is also a very high cases of MRSA infection compared to methicillin sensitive cases. And this is the erythromycin resistant cases among the Malaysian S. aureus isolate, also from 1990 to 2017. And it shows that there is a very high percentage of cases happens, and it has been reported by our National Surveillance of Antibiotic Resistant reports. This is the clindamycin resistance among the S. aureus isolates. Okay, so we also notice that there's a lot of cases happens in both private and government institution. So that also shows that antibiotic overuse and antibiotic misuse, it actually happens across the whole entire nation. So this is also the reason why that antibiotic resistant strain is actually increasing more and more year by year. So how do we actually have the emerging of the MRSA? MRSA basically they will develop when they are resistant towards a certain antibiotics by adapting themselves to the mode of action of these beta-lactam antibiotics. So when they develop the resistance, we will expect that this MRSA will have a low affinity for the penicillin binding protein. So this is a beta-lactam antibiotic. From here you can see if they are resistant, you can see that this beta-lactam antibiotic will no longer bind to these PBP2 sites, okay, anymore. Whereas currently, okay, we know that vancomycin is the drug that will become the last resort for any of the MRSA infection. But what we notice that MRSA strain is slowly resistant towards vancomycin as well. So that is actually has been emerging in many parts of the world. So this is why that there is a new, even much more resistant strain actually emerge. That is what we call as a vancomycin resistant S. aureus strain, the VRSA strain. So my interest is at looking at plant phenols. So based on the plant phenols, I'm interested to see that is there any plant phenols who are which is very effective in targeting those MRSA strain. The reason is because that nowadays we notice that synthetic drug compound is started to become not very useful towards attacking the MRSA anymore. So this is why that I wanted to look at the natural sources, whether is there any plant polyphenols are able to inhibit the MRSA effectively. So when it comes to phenolic compound, we notice that phenolic compound can be found in many different kind of plant-based food, like example, seeds, skin of the fruits, and even leaves of the vegetable. So all of these sources from the natural, they have a high concentration of this kind of phenolic compound. And not only that phenolic compound is something that exists in our food, we also notice that phenolic compound, they consist of many very useful biological activity, including antioxidants, antimicrobial, anti-cancer, anti-inflammatory, anti-aging, and they can also work as a preservative. They can also work as a neuroprotective agent, and also they can help in anti-diabetic. So when we look at the phenolic compound, coincidentally that few years back when I was looking at a few phenolic compound with a very close structural feature, what I noticed that there are three compounds that are very much similar with each other. You can see here we have the pyrrolgalol, very simple structure with only one benzene ring and three hydroxyl group, whereas another one is quite commonly exists is called the gallic acid. So gallic acid exists in many kinds of plant that we have in our surrounding, and also their derivative called the methyl gallic. So when you look at these structures of these three plants, you can see that they only differ in this side chain. So gallic acid, you have a carboxylic acid group here. Methyl gallic, you have a methyl gallic group here. Okay, whereas pyrrolgalol, there is nothing here. So that is the small differences that we notice among these three polyphenols. So when we look at the antimicrobial activity of these three compounds, we did a very preliminary assessment on the minimum inhibitory concentration and minimum bactericidal concentration. And other than that, based on the MIC and MBC, we also did a little bit of work using the HANQ assay. So we look at the pattern of the antibacterial activity from 0 to 24 hours. And at the same time, we also use the scanning electron microscope to try to observe the morphological changes that happens to this microbial, so that based on the morphological changes, we are trying to deduce, is there any possible target that this compound are actually working on the MRSA? Other than that, we also did a little bit of the work using the FDIR that is helping us to detect the cellular constituent changes in the bacterial cell. And lastly, we also did a crystal violet assay that looked at the membrane permeability alteration by the phytochemical compound that we have tested. So based on the MIC, we have noticed that the pyrrolgalol has the least MIC value. So the MIC value for pyrrolgalol is 15.6 microgram per ml, whereas gallic acid and methyl gallic, they are actually much more higher MIC. Like methyl gallic, MIC is 240 microgram per ml, whereas gallic acid, the MIC is 360 microgram per ml. So we notice that pyrrolgalol, this is a very small compound. Without the side group over here, they work even better than the derivative like gallic acid and methyl gallic. And interestingly, we found that methyl gallic with the methyl carboxylate group over here, they actually is much more stronger in terms of antibacterial activity compared to gallic acid, which consists of a carboxylic acid group here. So next, when we look at the gallic acid and methyl gallic, we notice that the antibacterial potency against the MRSA that increases with the alkyl chain length. So like what we have seen here, gallic acid only consists of a carboxylic acid group here, whereas the methyl gallic, they have a methyl carboxylate group over here. So what we notice that other than the MIC that we have just seen, when we try to observe these two compounds, the killing effect of the MRSA under the scanning electron microscope, we also notice that the cell actually lyses after the treatment. You can see over here, untreated cell, they are actually very round, very smooth. Okay, so this is how the untreated MRSA looks like under the scanning electron microscope. But when we treat the MRSA using methyl gallic, you can see that the cell will no longer like round like this. They have already like sort of like burst. Okay, same goes to gallic acid. The cell actually burst and they're actually clumping together. Okay, so that is the observation that we have seen under the scanning electron microscope. And next, we want to focus on the pyrrolgalol. How does it actually look like under the scanning electron microscope? And in the scanning electron microscope, which I will show you later how it looks like, based on the kill pattern of pyrrolgalol, we notice that when the MIC value actually increases, the killing pattern of pyrrolgalol towards MRSA is slightly differs a little bit. So when we look at the growth of MRSA under control, which means that there is no pyrogalo has been treated with this kind of microbial, you will notice that the lag phase of the pyrogalo, it will last for 4 hours. Okay, from here you can see that there are only 4 hours of the lag phase. So the same thing actually happens to the rest of the dosage that we are treated with pyrogalo. So the lag phase, okay, regardless that whether it has been treated with quarter MIC, half MIC, MIC, or double of MIC, the lag phase will only last for about 4 hours. After 4 hours, we notice that cells that has not been treated, they will have a maximum growth at 12 hours. So you can see that in the red line over here, there is an increasing in the number of microbial MRSA in 12 hours. Okay, after 12 hours, you will see that they will actually reach a plateau stage. But when we notice further on those cells which have been treated with MIC, we notice that lower MIC value will prolong the lag phase. So you can see over here, the purple, this is the green color. Okay, green color will have a longer lag phase. Same goes to purple color. So green color is quarter, purple color is MIC. So you can see that in MIC value, the lag phase from 4 hours, they prolong to 6 hours over here. Okay. And other than that, we also notice that there is a killing effect actually happen if you double up the MIC. Okay. At MIC, you notice that the lag phase has been prolonged to 6 hours. Then after that, the number of cells, they actually become lower and lower. But when you try to treat the cell with double of the MIC, that you will notice that after 4 hours, the cell actually drops. Here, MIC, it lasts until 6 hours. But for double of the MIC, the cell will actually start to drop after the 4 hours. So this is what you will notice over here. So from here, you can see that the MIC is actually equals to MBC because that the number or the concentration of pyrogalo, it actually works on inhibiting the bacteria. They are also started to kill the MRSA. Okay, this is a scanning electron microscope of the pyrogalo under the treatment of pyrogalo. So from here, this represents the untreated cell. This will be the treated cell. So untreated cell, like what I have shown you just now, the cells are very round, okay, very smooth surface, and they are just like those like small little grapes, and they will aggregate like those grape-like cluster cocci. Okay, there is minimal mucus on the surface of the cell. But when we treated this MRSA with pyrogalo, we noticed that over the morphological of this cell, there is a changes where we notice that the cell shape become abnormal, and you will see that on the surface of the cell, they started to become like pitted with some wrinkled surfaces, and some of them, they even have protrusion and heterogeneous appearance. And what we notice is that the cell number actually started to reduce. And when we look at further on the changes of MRSA membrane when we treat with pyrogalo, we also notice that there is a change in the bacterial cell membrane permeability. So as first, let's look at the control. So control, it is able to take up the crystal violet very well, okay, because that the percentage of crystal violet uptake is up to 70%. But when we treat the cells with quarter of the MIC, we notice that the cell membrane fluidity did not actually change much. Okay, so from here, you can also notice that the percentage of crystal violet uptake in quarter of the MIC, it actually not significant to the control. But when we try to treat the MRSA with the higher MIC, that is where you will notice that there is a significant changes on the crystal violet uptake. So like example, half of the MIC, the crystal violet percentage uptake has been lowered down to 60%. MIC, it is also almost close to 60, but when we double up the MIC, we notice that the crystal violet percentage uptake is only left with 50%. So this shows that the uptake of the crystal violet decreases with increasing pyrogalo concentration. that it is closely related to the severity of the membrane disruption with the pyrogalo concentration, which actually supported our prediction, what we can see in the scanning electron microscope. And when we try to proceed further to look at the changes of the bacterial cell using the Fourier transform infrared spectroscopy, FTIR. So in FTIR, a different region of the FTIR will give you some information on the changes. So like example, region 1, which is between 2008 to 3000, it will represent the cell membrane fatty acids. In region 2, from 1005 to 1007, they will give you an idea on the protein and peptides of the MRSA. In region 3, from 1002 to 1005, they will give you an idea on the changes of lipid, protein, carbohydrate, and nucleic acid, or even the phospholipid of the cell membrane. And in region 4, from 900 to 1200, it represents the polysaccharide or carbohydrate of the cell wall and cell membrane. And lastly, in region 5, from 700 to 900, it is representing the DNA and nucleotide. So based on this observation over here, you can see that region 1, okay, if the line is in blue color, that is representing the FTIR for untreated sample. Whereas the red one, it means the FTIR for the treated sample. So from region number 1, we notice that there is not much changes between the treated and untreated sample. So you can see that the pyrogalo is actually not working on the cell membrane fatty acid. But when we proceed to region 2, you notice that there is a reduction in the signal in region 2. So region 2 represents the protein and peptides of the bacteria. So based on the observation over here, there is a reduction in absorbance. So that means that pyrogalo actually works on the protein and peptides of the microbial. So that actually caused a reduction in absorbance in region 2. Region 3, same thing, they overlap perfectly, nothing happened. So we will omit that by pyrogalo is not affecting the lipid and protein, carbohydrate, and nucleic acid or phospholipid of the bacterial cell. Whereas when you come to region 4, another reduction of the signal. So that actually shows that pyrogalo is attacking the polysaccharide, carbohydrate of cell wall, and also the cell membrane. Whereas region 5, there is nothing happened in the FTIR over here. So based on the FTIR over here, we can conclude that pyrogalo is actually attacking the protein and peptides of the MRSA and also the polysaccharide, carbohydrate of the cell wall and cell membrane of the MRSA. So the important finding that we have noticed based on this study is that phenolic compound is a very good anti-MRSA agent that can inhibit and kill MRSA, and phenolic compound has the potency of killing the MRSA. So whether this phenolic compound, how strong they are in killing the MRSA, they are depending on the length of the alkyl group. So pyrogalo, the one that without any alkyl group, okay, they even have a better potency than gallic acid and methyl ester, despite that not having the alkyl group. So how did pyrogalo kills MRSA? They disrupt the cell membrane, cell wall of MRSA by attacking the fatty acid, protein and peptides, polysaccharide and carbohydrate of the cell wall of MRSA. And our work has been successfully being published in 3 scientific journals. So one, we published back in earlier, okay, in year 2018. One, we published it in 2020. And the latest one, we actually published the work in 2022. So all of these journals are actually very good impact research journals. Okay, so these are the references that I've used in my presentation today. So if you have any questions, feel free to unmute yourself and ask a question to me. Thank you.
Oh, participants, if you have any questions, kindly you are free to ask on by unmuting your mic, or you can post it on chat box also. Uh, Nobody is posting any question. I think
Thank
everybody
you so
is
much.
fine with your
Yeah.
understand and know whatever you are saying and happy to listen your
Mm.
findings. Somebody has asked, can we imply Tonda?
What do you want to imply?
See in the chat box, ma'am. Uh, Hani Kumar has given a question. Can we employ Tonda?
What do you mean by imply?
I don't know. Let me ask him to unmute himself. And
Mm. I saw one of the question is from Mohammed Shabi that I received it direct as a direct message. What are the major challenges, example like toxicity, stability in optimizing pyrrolgallo derivatives for therapeutic use? At the moment, Pyrrolgallo, even though it's a very good antimicrobial compound, but I noticed that Pyrrolgallo in terms of toxicity is actually quite high. So like example, if it is for ingestion, at the moment, I don't think it is safe for ingestion at the moment. But I would say that since Pyrrolgallo has been quite a good anti-MRSA agent, it will be much more wise at the moment with the current technology that we can use it as a sort of like example, you're trying to cleaning the surface, or you can develop into a disinfectant that work on surfaces. That will be actually much more safer compared to if you ingest your the Pyrrolgallo as a drug. Because this drug, even though the MIC is very low, but there is a toxicity issue which happen on this drug as well. So that's why that I would recommend that instead of using it for oral, maybe we can apply it as a disinfectant instead at the moment. In the future, if some research group they are able to solve the toxicity issue of the Pyrrolgallo, then it's actually a very potential agent to be used as an oral injection to fight against the MRSA.
somebody has asked, can we prepare any novel preparation for your work?
Uh, can we what?
Can we prepare any
Uh,
novel preparations for your work?
Not at the moment, not at the moment, because Pyrrolgallo is something still very new. Like what I mentioned, that other than the toxicity issue, which I actually noticed, and another issue that may actually happen with this compound is the oxidation. If we are putting this Pyrrolgallo into a formulation, there may be chances that this compound may be oxidized when there is other excipients that can react with this Pyrrolgallo. So this requires a further research on that. Because polyphenol compounds with a lot of OH group, there is a chances that they may react with other excipients present in the formulation. So that also requires a lot of work to do on it. I saw another from Mohammed Shabi, is a direct message to me. Can computational modeling predict the structure activity relationship for Pyrrolgallo-based membrane disruptor? I think you can. Not only that so-called like Pyrrolgallo-based, you can even try to work on the when you change the site group of the Pyrrolgallo or even gallic acid. Is there any changes in the binding towards the MRSA? I believe that it is doable. In few years back, I actually proposed this to one of my colleagues to work on it, because I feel that since the basic structure already given such a good activity, why not we try to modify the structure, putting different kind of functional group in other parts of the aromatic ring, and look at the binding and activity of this pyrrolgallo derivatives on the MRSA activity. I feel that it's worth to look into it. Okay, another question I received is, what ethical or safety considerations should be addressed when developing membrane active microbial? Of course, if we are developing a membrane active microbial, the safety issue is always the toxicity. Because regardless that whether are you applying this drug as oral or even topical application, you have to prove this is actually safe to be used in human. Okay, so toxicity is one of the things that we need to consider. Whereas for ethical, I don't think there is much issue on the ethical other than those ethics that you need if you are testing on human or animal. So in terms of ethical, it's still not that serious, because this pyrrolgallo is origin from plant. So there is not much ethical issue we can actually think of about this other than the human clinical trial and animal testing.
Ma'am, Mishra ma'am, Sujita ma'am.
Yes, sir.
Yes, ma'am. So now questionnaire
So,
session, ma'am.
yes, sir.
Okay, okay, okay.
People are directing their question directly to ma'am. So
Okay.
I was waiting if there is any other question
Right.
by the
Okay, okay.
participant.
Yeah, thank you.
Any more that you want to ask?
Does anyone else have any other question, or shall we move on? So we shall move on. I think there are no more questions. Ma'am, any question directed to you directly?
Another one from Muhammad Shabi again. Will natural plant-based molecule like pyrrolgallo inspire a new generation of eco-friendly antimicrobial? I would say yes, because nowadays that a lot of, not only that we can actually get a very good source of antimicrobial compound from plants, based on the structure of those plant-based compound, we can actually do some modification on the existing natural occurring polyphenols. We can also try to see that is there any specific functional group of those polyphenol is actually effective. Like what we can see that just now is that when we increase the alkyl group chain of the pyrrolgallo based compound, they actually increase the antimicrobial activity. So that's why based on here, maybe if let's say not on pyrrolgallo, maybe one day when we look for another structure of compound with a very simple structure, then maybe we can try to add different kind of functional group or even alkyl chain to the compound and look at the antibacterial effect on the bacteria itself. Actually, that worth for investigation, because there's a lot of like new knowledge that we are trying to discover every day. And currently that at the moment in the market, we know that having a drug which can actually resist those MRSA is something very useful. But the thing is that currently in the market, we are still relying on the existing drug because there's nothing much new drug actually come to the market in these recent few years. So this is why that when we are looking for a new drug to try to inhibit the growth of the bacteria, we would want to go back to the natural and see what is the structure of those natural products that we have and how was the activity in killing the bacteria in nature, because we believe that in nature there's a lot of good stuff actually present there, just that we need to have some time to discover them.
Ma'am, excuse me, ma'am.
Yes.
Is there any other target for this antibacterial activity? You are saying that one of the target, like pyrogalals, pyrogalal is one of the lead and pharmacophore. So we are targeting to the MRSA membrane. Okay, ma'am. So like that, any other target you could have find. So this is also one of the area we can focus our research.
Yes, because different kind of compound, like example, pyrrolidol, they target on the membrane. There are even some of the drug they are actually targeting on directly into the nucleic acid. Some of them even target the receptor. So different kind of compound, their target will be different. So depends on the pathway that these phytochemicals are targeting. Of course, how they actually target, we may have to spend a little bit of time to try to investigate. Like example, this pyrrolidol, initially we are not sure that what actually triggers this pyrrolidol to kill the MRSA. Then later, when we first started to look into it further, when we first looking at the scanning electron microscope, we noticed that there is a change in the cell morphology. That is what actually provoked us to look into further, look at the cell membrane, look at the FTIR, let's say cell membrane, because there are so many components present in the cell membrane. So what are the specific components that this pyrrolidol is actually targeting? So pyrrolidol, in this case, they are actually targeting on the surface of the cell. There are some other compounds we are actually targeting on receptor, some of them targeting on certain proteins, some of them are inhibiting certain pathway of the bacteria. So that's why it depends on what is the mechanism that each of these compounds are actually target. So cell membrane is only one of it.
So these, ma'am, when we are targeting to this MSRA membrane, so whether it is possible to kill the microbes of gram-positive, gram-negative, or other fungal infections, those things, really, which type of antibacterial action it will shows, like gram-positive or gram-negative, or both. So it's a broad spectrum like that.
Based on our previous experience in our research group in trying to finding the antimicrobial agent, what we noticed that gram-positive bacteria, most of them, those natural compound will target on their membrane itself. Okay, not only that, what we can see here, even like sometimes when we tested on the plant extract, which is having other polyphenols, they also target on cell membrane a lot. But of course, when we try to apply it in gram-negative bacteria, they may not work the same way, because we know that gram-positive and gram-negative, the cell membrane, the structure, how they actually form is actually very different. So when we look at the peptidoglycan layer of the gram-positive bacteria, from what we noticed that the peptidoglycan layer is actually a much easier target compared to the gram-negative bacteria, which doesn't have much peptidoglycan layer. So this is also one what we actually wanted to look into, that what kind of compound that it will only target on peptidoglycan layer and not on the gram-negative bacteria. So far, we noticed that quite a lot of the natural compound, they have no issue in targeting the gram-positive, but there's always an issue that it works on positive, but it is not working on gram-negative bacteria.
Positive.
So that's why
Yeah.
when we look at gram-positive, instead of looking at the original, the simple normal strain, this is why that we wanted to look into the antibiotic resistant strain, because antibiotic resistant strain, we know that there's not much drug we can use to treat them. Okay, so that's why we are interested to try to use some natural compound in looking into the antibiotic resistant strain.
Ma'am, basically, your work will be starting with in silico studies, or else directly you are going for the natural product testing on the.
I did not start with in silico. This finding is actually, I would say that it wasn't being planned like from in silico to in vitro, all those things. This study actually began when I was doing the isolation. So I managed to isolate the gallic acid and methyl gallic. So when I look at the structure of the compound, they actually gave me an idea that actually this side group actually plays some role in the antibacterial activity, because based on the MIC itself, we see that there is a differences of the MIC. So when I was actually wondering that, what if, like, let's say if they don't have the alkyl chain present there. So that is when I tried to study on Pyrogallo. So Pyrogallo, surprisingly, it gave us actually even much higher antibacterial activity despite not having that alkyl chain. So that is actually what actually caused us to go into this research further. So we did not plan from in silico and then go into the in vitro. It was started off with some accidental finding, and then we try to look for some new knowledge from there.
Okay, so ma'am, basically this pyrogalal, the pharmacophores, where it is present, ma'am. So like what type of natural products which contain these things?
Actually, a lot of the plant we have the pyrogallo, and even sometimes that when we are doing the isolation, we notice that inside the plant itself, there may not be in the form of pyrogallo, but throughout the isolation, there may be some processes. The phenolic acid actually is being hydrolyzed to become pyrogallo. That is what we actually can see in our MS when we do the isolation. That originally from previous isolation, what we can see in MS is the gallic acid. But during the isolation, some hydrolysis actually could happen. Then in the later stage, we notice that we're actually getting a pyrogallo at the end.
Dean sir?
Yes, ma'am.
Sir.
Yeah. We can conclude the session. Yes, ma'am.
Yes, sir. Thank you so much, ma'am, for such an
Yes, thank you.
insightful and thought-provoking session, and very patiently replying to all the queries you have got. Now, I will like to invite our Dean sir, Dr. Rahman, for the virtual certificate of appreciation to respected speaker, Dr. Chu Yik Lee, and share his words of encouragement.
Yes, ma'am. Yes.
Thank you, sir.
So,
Thank you,
ma'am,
Prof. Rahman.
ma'am, Chiu Ling, ma'am. So I am very much thankful and grateful. Like you have accepted our consent, and immediately you have accepted our consent, and you have delivered no new area of research to our faculties. So this is a faculty development program. So these 5 days journey, each day, one or other topics are different. So some of them no delivered lectures in the in silico, the drug design and discovery, nano herbal formulations, then nano drug delivery systems, Sutix related, and like medical devices related, nervous system targeted drug delivery. So like that, 5 days, 5 different areas we have designed the topics, and we have invited the speakers. Three of them foreigners from 2 to 3 members from India, like ma'am. So here today also we had an excellent international exposure to our faculty delegates. So really it's a awesome ma'am, and we are kindly accept our this online thing, and I will share you the certificate of appreciation through this one. Uh, Thank you for spending your valuable time. Basically, you may be busy with your regular academics and administration related activities. However, you have accepted our invitation and joined with us. Okay, ma'am. So, ma'am, hope we can collaborate, because I am also basically chemistry person, medicinal chemistry and pharmaceutical chemistry. My core research area is in silico drug design and discovery. Then nano naturally related works also I am carrying out basically. Okay, so anti thyroid activity, anti-thyroid activity, then gastric related things. So 2 3 areas I am also working with my PG and PhD students. Okay,
Mm.
so whenever this opportunity comes, we can collaborate, and in terms of the research and publications, you also please guide and motivate our students. Okay,
Sure,
ma'am. Yeah,
sure.
correct. So once again, ma'am, I am very much thankful, ma'am. Thankful and grateful.
Thank you. Thank you for inviting me for this EFDP.
Thank you so much, sir. Thank you so much, Dean sir. As today marks of the final day,
Yes,
fi- uh, final day of our FDP, I would like to acknowledge the wonderful session we have had over last 5 days, from drug design and delivery strategies to cutting edge analytical techniques and phytomedicine research. Also, the database that Gaurav sir has gave us, that was also very enlightening for us. Each session added immense value and contribute to the academic investment for all the participants. Um, Before conclusion, I would like to express my heartfelt gratitude to everyone present here. Over the last 5 days, this FDP has not only brought together experts from the all around the world, but also have created a platform for learning, discussion, and collaboration. The enthusiasm and curiosity of all have shown a reflect true spirit of academic growth for all my participants, all those 200 and above participants which we are getting from last 5 days regularly. Uh, to our esteemed speakers, thank you for sharing your time, expertise, and insights. To our organizing team, our dedication makes this event seamless and impactful. All the participants to actively involvement and gave life to this FDP. Thank you everyone for participating in this. We all bring this program to a close. Let us carry forward the knowledge and gain the ideas, exchange the inspiration shares. We may these learning help you in innovate, explore, and contribute meaningful and encouraging and innovative ideas in the field of pharmaceutical sciences. Everybody, feedback section of day 5 will be published in the Zoom meeting. So you can fill the form of feedback for day 5 also, and your certification and everything will be uh, emailed to you within 1 week of the ending of this FDP. Thank you once again for being a part of this FDP. Wishing you all a continuous success, good health, and bright academic journey. Have a wonderful day and see you all in the future for academic endeavors.
okay.
Thank you so
So,
much.
ma'am, thank you, Suchita Misra, ma'am, for your excellent hosting these 5 days, right? And We are also
Thank
thankful
you
to
so
you,
much, sir.
thankful to you, Sujita Misra ma'am, for coordinating all these international FDP without any disturbance and without any defaults of the sessions. Okay, so we are thankful and we congratulate you for your no sincerity and hard work, hard work.
Thank you so
And
much, sir.
coming to the faculty delegates, I request everyone fill out the feedback of day 5 also, and we will consolidate and take the average of all the 5 days attendance. So based upon the attendance, we would like to release the certificate to all the faculty members, those who joined in the FDP program, right? So it doesn't mean ki one session we have attended and expecting certificate, it is not correct. So so there should be a cutoff mark and cutoff attendance percentage. So based on that only we will release the certificates to the faculty members, right? So that's why once again I request today's day 5 link also please you fill out, and the average of these 5 days attendance will be taken into the consideration. Then we will release the certificate to the faculties. Okay. So once again, no, I am firstly very much thankful and grateful to Chief Ling ma'am for in time joining and for excellent and informative session delivered to our faculty delegates. And thankful to all of my team, Galgotias University faculty, the Department of Pharmacy, School of Medical and Allied Sciences, and special thanks to hosting team, Ms. Ichita Misra, Mamata Vishnoy, Neha Sharma, etc. Hope we will come up with a new idea and a new event. All the delegates in future also you please join the same. Thank you, ma'am. Thank you, Chief Ling, ma'am. Thank you very much, ma'am.
చాట్ బాక్స్ కనిపించలేదేంటి?
So anyone wanted to speak anything?
Yes, sir. This is
Ah,
Dr. PHS Kumar speaking, sir.
yes, Pani sir, tell me, sir.
I'm from Aditya School of Pharmacy,
Okay.
IIT University.
Ah,
Sir, you have conducted the very beautiful sessions,
thank you,
so
sir.
which are very useful for us.
Okay,
So we
sir.
have lot of doubts on novel drug delivery system. What
Okay.
I feel that most of them have resolved with this webinar. So
Okay, sir.
in future we are expecting such a kind of beautiful sessions from you, sir.
Okay. Thank you very much,
Uh,
Pani sir. Thank you very much, sir, for your no, good words to us. And I also
Thank you, sir.
request any other faculty delegates would like to give your inputs. Please give us. Any other faculty delegates? If you want to share your views regarding this 5 days MDP, you can always welcome and share your views.
Good morning, sir. This is Gayatri from Malaradi College of Pharmacy, Hyderabad.
Yes, good morning, ma'am. Yes, ma'am.
Sir, actually, first 2 days the link I didn't get proper that one, but after 3 days
Okay.
I have attended. All the
Okay, ma'am.
3 days sessions are very nice, sir. First one,
Okay, ma'am.
actually I am from chemistry department.
Okay.
So drug development using different tools. Yesterday the session is very nice
Yes,
because
ma'am.
the whatever the FDP you have taken, the concept whatever you have taken, that is different from all other FDPs, sir. Till now, whatever I have attended.
Okay, thank you, ma'am.
So very much impressed about your FDPS, sir. Seriously.
Okay, ma'am. Yes, ma'am.
Okay.
Thank you very much.
Thank you so much. I hope based on see these criteria only again you will go for the FDP. Again you will select a new innovative topics. Thank you so
Yes.
much, sir.
So actually, ma'am, we are expecting CM from the faculty members because So for accreditations, this is basically important. So how many faculty members attended the faculty development programs like that. So I could notice so many faculties, just they will open their Zoom link and joining the session and doing some other works. If you can spend this minimum of 45 minutes or 1 hour time, so you can acquire some skills from the experts. So what
Seriously,
research,
sir.
what
Yesterday
present,
means
what,
whatever the FDP you have taken, that is
yes,
good because completely means yesterday nanotechnology, whatever the equipments or whatever how we can approach the nanotechnology means for herbal drugs and
yes,
also the drug delivery systems.
yes,
So
ma'am.
each and every topic, whatever you have taken, that is means completely different from the previous FDPs where I have attended.
Yes, yes, ma'am. That's what I am
Thank you
telling.
so much. Very
This is,
informative session, sir, for FDPs. 3 days. I have attended actually only 3 days because first 2 days I didn't open. I
okay,
can't
okay,
open the link due to network issues.
okay, okay,
But
understood.
whatever the 3 days I have attended, it is a very good session, sir.
Right, right. Thank you very much, ma'am. Thank you, Gayatri, ma'am. Hope we will give best in coming days also.
Okay, sir. Thank you.
Thank you, ma'am. So if any other faculty delegate would like to share your views, please. And we think all the sessions on time we have started. So it means we said 11 o'clock and 11:05 we have started the sessions, daily basis without any, glitches and the networking and all we have conducted. And day one with the so the gracious presence of Vice Chancellor Sir of Galgotias University and Deputy Vice Chancellor of Cyber Jaya University and Director of Research, Cyber Jaya University has joined for the inauguration session, followed by so other sessions we have successfully completed. So any other delegate would like to share your views, please. Okay, so hope I think everyone received the day 5 link. So please fill out the day 5 the feedback, and we can release the certificates after the 1 week, because certificate has to sign by Vice Chancellor of the Galgotia University and Cyberjaya University also. That's why it will take a few days delay. However, I request all the faculty delegates, you know, so don't worry, we will reach out with you the certificates. Okay. So because two vice chancellors have to sign. This is the international FDP. So then only our certificate is highly valuable, right? So once again, thank you very much for everyone for joining with us. Ma'am, thank you, ma'am. Ma'am, thank you, ma'am.
Okay,
Yes,
sir.
Thank
ma'am.
Thank
you.
Yes, ma'am. Thank you. We are closing
you.
down the sessions.
Okay, sir.
Okay, thank you.
Now, every delegate, I have posted the feedback link in the chat box as well as in the chat box of the YouTube link also. So everybody can access that. And now I'm ending the session.
Ma'am,
As
ma'am, a small.
Yes.
Actually, due to network issues, I can't connect first 2 days. So
Gayatri, ma'am, no?
that's what did.
Gayatri,
Yes,
madam.
sir.
Yes,
ma'am,
yes.
whoever is facing such
Yes,
kind
sir.
of
Yes,
problem,
sir.
Yeah, we will
they
check
can
it out,
reply
madam.
to they can reply to the mail which we have sent the link in. Okay, the joining link which we have
Yes,
sent.
sir. Yeah, ma'am.
You can
Actually, I
reply
have
to that
replied.
mail.
Okay. Ma'am, we will take care,
So if
ma'am.
you will
Gayatri, ma'am.
reply there, I will send the link to there. Okay,
Right.
Okay, sir. Okay, ma'am.
Right,
personal
ma'am.
Thank you
Right.
so
message
much.
you
Okay.
do not reply in that. Do a personal message on that.
Okay.
Okay, ma'am.
Okay, thank you.
Thank you.
Thank you, sir. Thank
Thank
you,
you.
ma'am, once again. Now